Good day, everyone. This is Jack Van Horn from the USC Mark and Mary Stevens Neuroimaging and Informatics Institute, the home of the BD2K Training Coordinating Center, and I'd like to welcome you all to the Big Data to Knowledge Guide to the Fundamentals of Data Science webinar series. We are very fortunate to have Dr. Daniel Marcus from Washington University in St. Louis today, who will be discussing imaging informatics. Dr. Marcus is a PhD in neuroscience from Washington University, which he received in 2001, where he remains as an Associate Professor of Radiology, and he directs the Neuroimaging Informatics and Analysis NIAC Center, as well as the Human Connectome Coordinating Facility. His expertise lies in the area of neuroinformatics, including the development of data management systems, ontologies, and visualization tools. He's been a longtime advocate for the open data sharing effort, which he practices through the Human Connectome Project, through other projects such as OASIS, and another one called XNAT Central. His lab leads the development of the open source XNAT Imaging Informatics Platform, and we are very excited to have him here today. I want to invite all of our listeners to please submit any questions that you have for Dr. Marcus during his presentation via the GoToWebinar question submission system, and we will sift through those and get answers for you on the most salient ones at the end of the hour. Without further ado, please allow me to turn it over to Dr. Marcus for his presentation on imaging informatics. Thank you so much, Dan.
Thank you, Jack, for the introduction. And welcome everyone who's joined today, and appreciate those who've been able to come. So here's kind of how I thought we would do this today. I'd start by talking about why imaging is important and what kinds of imaging are in play for the kinds of informatics I'll be discussing. And then I'll give kind of like a didactic kind of background on imaging informatics infrastructure as used in the clinic. And then talk about some of the imaging informatics infrastructure that we've put in place for the research enterprise and how those two kinds of informatics interplay with one another. And then I'll talk a bit about some of the imaging informatics tools and systems that we've built to do data sharing, and a little bit about how informatics can support translational research. And then finally, if I have time, and that might be a bit of a stretch, I'll talk a bit about how you can actually make a living doing imaging informatics, and it's something I've been doing for almost 2 decades now and really enjoy, and I think a lot of other folks would find a lot of value in it as well. Okay, so why imaging is important to begin with, and why we might want to consider building imaging informatics tools to support it. So there's, of course, lots of different kinds of imaging that cover a very, very broad spectrum, many orders of magnitude of scale, and that have very different kinds of preparations and apparatus to acquire the images. What we're going to be talking about today is primarily imaging at the millimeter and submillimeter scale, and includes the many of the common medical imaging modalities. Which of course isn't to say that the other kinds of imaging aren't important, but there's a lot of reasons why the medical imaging modalities are of particular interest. One, of course, is that they're non-invasive and non-destructive, so that they can be done both in healthy humans as well as in humans who are being imaged for diagnostic purposes. And because it can be used in healthy people, it means we can use it for research and in healthy research participants. Also, this kind of imaging can be done in humans at a time scale that they can tolerate. So typically in less than an hour, you can acquire a lot of very interesting imaging. These modalities also allow us to look inside the organism. That is, we can see organs and other tissue below the surface, so we can actually see what's going on internally. And we can see not just the structure of the tissue, but we can often image various aspects of the function, including things like metabolism, or in the brain, the Bold signal, which allows us to understand how the brain might be functioning. We can also apply these methods across species, and in particular, we can work with mice and other rodents to develop the methods that then can translate into humans. And these same instruments can be used in the clinic, but also then in research. So some of the commonly used imaging methods include radiography. So of course, if you go in and get an X-ray, computed tomography, which gives us something along the lines of a three-dimensional radiograph, magnetic resonance imaging, which includes anatomic images, but various kinds of functional images as well, including fMRI, diffusion, perfusion, and spectroscopy. And then there's nuclear medicine, modalities like PET and SPECT that allow us to see, again, various kinds of functional things going on inside the body. There's other kinds of clinical imaging, of course, things like pathology, and those tend to have different informatics requirements, and of course they're very important, but a little out of scope for today's talk. And then there's a lot of non-clinical kinds of imaging like MEG and OCT, which again are very important and may eventually become clinical modalities, but currently are largely not, and therefore a lot of the informatics infrastructure is quite different. And of course, there's lots of other kinds of imaging at other scales that again require other kinds of informatics capabilities. Okay, so let's talk a little bit about the kinds of informatics that is in place in the clinic to enable images to be collected and accessed by clinicians. And this is kind of a nice figure showing the overall kinds of workflows used in the clinic. A lot of it starts with the RIS, the Radiology Information System, which is used to schedule the patients and to identify what exams they're going to get. Once a patient has been scheduled, then an exam can be requested at the modality, in other words, called the scanner. And once the images are obtained at the modality, then those images are transferred to some sort of data system using the DICOM protocol. We'll talk quite a bit about DICOM here in the coming up slides. Within a clinical environment, the images are typically sent to a PACS, and they're sent over the DICOM network protocol. The PACS is the thing that actually stores the images. So this is like the big image database. When a clinician is ready to view the images, they're transferred to a viewing workstation over the DICOM network protocol. And it's at the workstation where the radiologist does his interpretation, and the results of that interpretation are sent typically back to the PACS and the RIS systems. All right, so we need to talk about the DICOM standard. This is the Digital Imaging and Communication in Medicine, although everybody just calls it DICOM. And this is the global standard produced by the organization called NEMA. And you know it's a standard because it took about 20 years to develop and covers about over 6,000 pages of specifications. It's so complicated that vendors are required to produce a conformance statement describing how they actually comply with this complicated protocol. And you can read through those conformance statements and really get an understanding of what portions of the overall DICOM standard that a particular device implements. One of the nice things about DICOM is that it really is a standard in that it's been universally adopted by virtually all manufacturers and for all modalities used in the clinic. Interestingly, the standard includes a definition and specification of both the data format, so how the images are stored, as well as the network protocol for communicating those images between systems. It includes both the acquired images as well as various kinds of derived resources. So something, for example, like a structured report or a tractography map can all be stored in DICOM objects. It's a standard that is ongoing in development to support new modalities, new communication protocols, new data structures. It tends to be a slow and laborious process for new capabilities to be added to DICOM, and that can be often to the frustration of those of us on the research side who need things to move a little faster, but is somewhat necessary for clinical communications. So for the file format, it includes a number of different components, both the pixel data itself as well as a substantial amount of metadata about how the images were acquired. DICOM does have kind of its own terminology that's useful to get used to. It refers to the person who's put in the scanner as a patient, of course. But the images that are acquired, the overall set of images for a particular patient is called a study. And the individual acquisitions are called a series. So those of us on the research side often call these things, the patient we might say is a subject or a participant. Study we might call a session or just a scan. And then the series, sometimes we call those just the image, or we'll call them, sometimes those the scan. So there's on the research side, there's a lot of kind of mixed and overloaded use of these terms, but they're pretty strictly used within the DICOM side. There's a whole lot of metadata included. So this image here actually shows a tool developed in my lab called DICOM Browser that allows you to open up a DICOM image and inspect all of the metadata. And you can look through this and find really helpful things about like what date the image was acquired, lots of the particular settings at the scanner. So for an MRI study, you would have things like the repetition time and the flip angles and all of those kinds of things that are very detailed, low-level information about how the image was acquired, and that can be quite helpful for doing downstream processing. It's also important to note that there is a lot of PHI potentially embedded, patient identifying information embedded within this metadata. So for example, there is the patient name. In this case, we've already anonymized it and just call them sample patient. But often the data coming off of the scanner will actually have the patient name included. And to make things more complicated, it's the patient identifying information is often included in fields that are as obvious as patient name. But there's more subtle fields included that can be quite difficult to identify whether PHI is included or not. And to use the data on the research environment, a lot of effort has to go into identifying all of that lurking PHI. And to complicate matters more, depending on the modality, the PHI can actually be embedded in the pixels themselves. So you'll often find, for some modalities, ultrasound is an example, where there's actually the patient name burned into the image itself. And you have to use tools that actually can go in and edit the pixels to remove that identifying information. So here's just a blown up view of the DICOM browser. The DICOM networking, so this is how when you have a DICOM file, it's moved around across various information systems. There's a number of kind of unique terms that are used within the DICOM standard to describe how the images are moved and how queries are made. The actual movement is done over TCP/IP using very specific DICOM protocols that have different names. One is C-store, which is actually kind of the most obvious of the ways of moving files, which just says take this data from a scanner and store it into some sort of storage device. So for example, the PACS, which as we mentioned before is the imaging database. But then there's communications like a C-find, which is to submit a request that a tool is interested in cases that meet a particular set of criteria. So that might be an accession number or a study date. And then the system, the PACS, for example, would send a response back to whatever tool is asking for images that meet those criteria. So that's the C-find response. When the actual tool is interested in getting the images after it's done a C-find, it submits what's called a C-move request to get the images, to tell the PACS to send the images, again, using the C-store protocol, over to the viewer. So it's somewhat of a kind of circuitous way of getting the images instead of just saying, pull the image from the PACS to the viewer. You're instead saying, hey, will you push those images to me. So it's a little bit complicated and takes a little getting used to. This feels, when you're used to doing web-based kinds of communication, this can feel like a very weird and sort of tedious way of communicating. But those in the DICOM community have gotten pretty used to it. But nonetheless, they've been responsive to those of us who've been asking for more modern networking protocols, and have implemented a thing called DICOM Web, which provides RESTful web services for doing these same kinds of communications. It's a relatively new part of the DICOM standard, and so it's not really broadly adopted to date, but hopefully will be coming in the coming years. There's a lot of really handy DICOM tools that are out there that those of us who practice imaging informatics use on a daily basis. These are some of my favorites. It's certainly by no means a comprehensive list. We use DCM for Shea, this top PACS in my lab quite frequently for a lot of our DICOM infrastructure and capabilities. It's a nice open source Java tool. DCMTK provides very similar kinds of functionality, but using C. And then there's tools like Orthanc and Clear Canvas, which are really kind of turnkey PACS systems that are open source and freely available. We use both of those frequently in the lab. Clear Canvas in particular is a nice Windows-based PACS, includes both the server side as well as a viewer. One of the big challenges in handling DICOM for research is making sure that the data are properly anonymized and to be able to inspect the images. We often use a tool called the Clinical Trial Processor. It was developed by RSNA and is a web-based application to do a lot of different kinds of DICOM manipulation and transferring of those images and routing of those images between systems. The screenshot I showed before is DICOM Browser, a tool built in my lab, which is just a nice handy desktop application to be able to quickly review metadata tags, to manually edit them and transfer the images to a DICOM storage system. For viewing DICOM images, there's a lot of useful tools out there. OSIRIX is very widely used. The OSIRIX brand name is includes a commercial product, but there's an open source version of it called HOROS. It's a Mac specific application, but very widely used in the Mac community. 3D Slicer provides a lot of DICOM functionality that can be fed into a very sophisticated set of analytic tools. And then OHIF is producing a web-based DICOM viewer that has a lot of capabilities, and the advantage of being entirely web-based. You don't have to install anything on your desktop. All right, so that covered a lot of the kinds of data flows and standards and networking that are used in the clinic. And those of us who mostly do research, it's very important to be aware of those, because that is the level at which we need to interface with those systems in order to get our images or to submit information back into clinical workflows in translational contexts. But I'm going to switch now to imaging informatics tools that we use really explicitly in research that have DICOM capabilities and know how to talk to the clinical infrastructure, but are really more focused on research types of operations. And if we think about really what are the differences between clinical capabilities and research capabilities, they are somewhat different. So in the clinic, everything is really about the patient. So a doctor needs to see a particular patient, their current exam and prior exams, and they need to be able to annotate that patient and communicate diagnosis to other physicians. On the research side, yeah, it's important, the individual subject, but we're also really care a lot about group level operations. So things that are done at the project level and include analyses that are not just of a single patient, but of large groups. The clinic is also burdened by FDA regulations, which are, of course, very necessary to protect patients, but are quite restrictive in the kinds of things that you can do with the systems. In particular, you can't just code new functionality on it whenever you want. Versus on the research side, we need more agility because we're really trying to be at the bleeding edge of things. And so we need to be able to easily adapt and incorporate new functionality. Clinic obviously needs access to all the identifying patient information, whereas on the research side, we explicitly don't want identifying patient information. And those can often be kind of in conflict with one another for supporting interactive workflows. On the clinical side, it's important, obviously, to be able to link with the more broad medical record. On the research side, we're interested in linking with potentially very large amounts of non-imaging research data. So things like behavioral assessments or case report forms or genomics. The clinic is often more of a qualitative type of practice where a radiologist is providing their assessment of images by observation. On the research side, we tend to use a lot more automated and semi-automated quantitative analytics. And those are often quite computationally demanding. In the clinic, doctors share the data with one another, or the images with one another, in a somewhat open way, right? Like, so if you're in a hospital, the assumption is any of the doctors in the hospital are privileged to be able to access those images. So security is typically done at the level of restricting who has, within a hospital, has access to the system and making sure that people outside the hospital don't. Versus on the research side, we're often sharing more selectively with our collaborators and then eventually with the community. But as we, that data sharing follows sort of a progression of a broader and broader access as data goes through the research process and publication. So considering these sort of differences, we can think of a workflow on the research side that looks something like this, where we have the data coming from different systems, including the scanner, but also potentially other kinds of measurements, genomics, case report forms, and the like, and other sorts of data systems. And then the data kind of goes through a controlled research process where you have individual researchers who are working with the data. They work then with their collaborators who also want access to the data. And eventually, if everything goes well, the investigators share their data with the broader community. But this needs to be a pretty controlled process in order to be able to kind of secure access to the data and have the right controls over it as it makes its way through the research process. So we've developed in my lab a system called XNAT, which provides this sort of controlled process for working with research data. So this is what XNAT looks like from the web interface. This is a particular XNAT instance that supports the Human Connectome Project. But XNAT is an open source tool that anybody can download and install and run as their own informatics platform. It provides a broad suite of features for archiving data, managing it, running analytic routines, visualizing images and doing markup on images, and then sharing that data with collaborators and the community. It is open source. It has an open RESTful programming interface, so you can integrate external tools with it. And it's pretty widely used around the world. Originally, because I'm a neuroscientist, we supported primarily neuroimaging, but over the years, we've really made strides to make it a very useful tool for really all imaging endeavors. We think of it as a platform because it's built to be very flexible for lots of different use cases, including clinical and translational research, large institutional repositories, multi-center studies, and open access data sharing. And I'll go over a few examples of those different kinds of use cases as we proceed here. So this is the XNAT architecture in some detail for those who like kind of the technical view of things. Importantly, it does have a full DICOM suite of services, so it does know how to communicate with DICOM workflows from the clinical side. So you can push images directly from modalities. You can query out to the PACS, and you can submit data from XNAT back into PACSes. And that's really useful on the clinical side of things and integrating with the scanners themselves. But most of the interfacing with XNAT is through its RESTful API. So lots of other external applications, visualization tools and things like that can communicate with XNAT through using kind of more modern web communication. I'm going to be talking, a lot about XNAT because it's the system I know best, but it's important to note that there's actually quite a few different informatics systems to support imaging research. Here's some of them. COINS, LORIS, and Flywheel are primarily used within the neuroimaging community. MIDAS is pretty broadly used within the 3D slicer community. MRIDB is an MRI-focused database. NBIA is developed by the National Cancer Institute and is the system that underlies the Cancer Imaging Archive. So there's a lot of these tools that are out there, and they have overlapping but distinct feature sets. So if you're thinking about deploying a system like this, it's really useful to kind of review them, try them out, and see what's the best fit for your particular project. At this point, there's still a lot of labs that have chosen to kind of build their own and kind of glue together various components, largely because there's legacy infrastructure already in place at a center. But a lot of these tools are getting to the point where they're pretty turnkey, they're very powerful, very feature rich, and they're open source, so you can add to them. So I would suggest, considering not building your own, but using one of the tools that's already available, and to consider engaging with the open source development community around that tool. So back to some of the things that we're doing in XNAT. As I mentioned, there's full DICOM integration, but there's a lot of other stuff going on, and I'll kind of go through some of those in some detail here in a minute. The way we organize data within XNAT overlaps or looks kind of like the organization of DICOM, but is not completely the same. So we organize data within a project. Any data that's added to the system has to be within a project, and that's the level at which we do all the user access control. And a lot of the kinds of operations that are done are configured at that project level. Within a project, you have one or more subjects. These are the people who are going in to get experimental data. And each subject can come in for one or more visits. So you might imagine that they come in for something like a baseline visit and then a 3-month or 12-month follow-up. And within a visit, you can have different kinds of experiments. So maybe they come in for a baseline and they get an MRI and a PET exam, as well as some kind of clinical workup. Each of those kinds of things would be considered an experiment. And then within imaging experiments, you can have one or more scans. So this would be in an MRI, for example, you might get a T1 weighted MP range. But you can have multiple scans. You might have a dozen different individual scans acquired within a single imaging session. And that data organization is reflected in the web pages that XNAT provides. So there's a project level page that gives you a lot of information and group level details about everything that's happening in that individual project. If you are an owner on that project, you have various kind of administrative features on the page that allow you to control who else has access and what kinds of operations are configured for that particular project. If you drill in on an individual subject, then you can see the details of what experimental data has been acquired for that subject. So this subject, for example, has come in for an MRI visit. They came in for a separate MRI visit. And within each of those visits, various kinds of additional data have been included in the system. So there's been quality control reviews and a DICOM review validation for each of those visits. You can drill into a particular MR session, and then you can see the real low-level details of exactly what was acquired within that session, and you can expand each individual scan to get the very low-level details, most of which are extracted from the DICOM when the images are imported into the system. So there's a number of ways to get data into XNAT. The most frequent one is just using that DICOM capabilities that are already built onto the scanner. So from the scanner console, you can just hit submit and send it to a XNAT receiver that's been configured at that scanner. So this is really useful for archiving from your local scanners that are on your network. DICOM itself is not has typically implemented a secure protocol. So typically you don't send DICOM from remote scanners to over DICOM protocol into XNAT. XNAT then does, as the data come in, it imports the data, figures out what project it's associated with based on metadata tags in the DICOM, and then puts the data into the correct project. It does have an anonymization scheme that I'll go into shortly for removing PHI and typically replacing it with research identifiers. If you are in a situation where images are being acquired remotely, there's a couple of different options for how to get it into XNAT. One is to set up these little relay devices. So these are little Intel NUC boxes, and you can actually install kind of a lightweight version of XNAT on one of these boxes, and then hook that up to the scanner, submit to DICOM using the DICOM protocol into the device, and then it submits over HTTPS using XNAT's RESTful API to archive the data in a remote XNAT system. So you have going into the device DICOM, and then coming out of the device is secure HTTP to move it over to the remote system. And that's really good if you're going to have a lot of data acquired at a particular site. For less frequent sites that are maybe just collecting a patient or two a week, they can come to the website and just do a manual upload using tools that are included directly on XNAT. In all cases, the same anonymization profiles are executed on the images, and importantly, it's done before the images are transferred from the study site. So the remote system never sees that identifying information. That identifying information never leaves the local network. XNAT also supports doing query retrieve from the PACS. So if the hospital, once you've worked with the hospital IT to allow connecting to the PACS, you can submit queries to it using things like patient name or accession number, and find patients of interest that then get pulled into dedicated projects on your XNAT system. So this is really useful if you have patients enrolled in a prospective study that are getting standard of care imaging that's going through the regular patient workflows, but you have them enrolled on the study and have IRB approval to add those images to your research database. So the DICOM anonymization that's built, language that's built into XNAT looks like this. It's actually looks a little complicated, but it's pretty straightforward. For each of the DICOM metadata tags, which are typically identified by these coded numbers, you can do different kinds of operations on them. So in this case, the operation is to replace whatever's in that tag 1010 with the value subject 346. Here it indicates to remove this entire tag from the file itself. So it's the birth date in this case would be just the tag would be gone. Here you were doing some more complicated logic where it's saying, if I find in this tag the value CCIR3T, then replace it with this value. And there's quite a bit of different complicated logic that can be done in order to achieve the kinds of anonymization that are necessary to handle potentially very complicated situations. XNAT does have a full user role and permission system so that you can really, at a very fine-grained level, control who has access to what data in the system. So you could, for example, build a user role called MRI reviewer and assign a person that role on a project, and then they would be able to review and see MRI scans, but wouldn't be able to see any other imaging modalities that might be part of that project. So you can get very fine-grained on exactly what kind of data they have access to, and then what privileges they have with that data. XNAT also embeds case report forms or data entry forms so that you can collect data other than the imaging. So for example, if you're doing a neuroimaging study and are using the Wechsler IQ scale, you could have a form to capture the measurements from that assessment into XNAT. There are already many hundreds of forms that have been built into XNAT, and many people have shared those with the community. It's also pretty straightforward to add your own if you have a custom form that you need to implement. XNAT also supports a number of quality control procedures, and this is really critical in the research workflows because it's so easy to accidentally acquire the wrong acquisitions, or for the settings of the scanner to be slightly off of your expected acquisition protocol. So one of the forms of QC that we have is to actually review the DICOM fields as the DICOM is being imported. We call this protocol validation. So using a little specification file, you can tell the system exactly what you're expecting within the metadata. In this case, we've set all of these expected values for all of these fields. Most of them pass, except for the phase encoding rotation was slightly out of the expected range. So when a session is uploaded into the system that fails the validation, an email is sent to the research team so they can very quickly go and review the images and potentially even rescan the subject while they're still on site. System also supports doing more manual review of the images to look for things like artifacts or poor head positioning. The exact QC procedure that you use can be configured in the system so that it captures really your very specific kind of review process. And then you can do a more automated review of actually running routines to check the images for various characteristics. So in this case, we're looking for motion artifacts in an fMRI scan, and you can see for this particular subject over time that they have a number of events where they're moving quite a bit. So this chart would indicate a subject who was really moving a lot in the scanner may need to be given instructions to hold still and rescan. I'd mentioned that XNAT has a RESTful API, and it's really a very powerful way to expand XNAT's capabilities, in particular by sharing data from XNAT into various kinds of applications. So in this case, we've written a plugin for 3D Slicer that can be opened up, and you can type in your credentials and what XNAT you want to talk to, and then work through this dialog to select cases that you're interested in. Then those open up inside of Slicer, and you can do various manipulations, draw regions of interest and the like, and submit those results back into XNAT, where they'll be stored alongside the original images. So we have that for 3D Slicer, we have it for OSIRIX, and for many other applications. A nice thing that a lot of sites are doing now, because they are sharing data between different organizations and have lots of kind of complex workflows, is to be able to do synchronization between two XNAT systems. So that we have a plugin that allows you to define what systems to send, what other XNATs to send particular exams to, and the plugin takes care of a lot of that communication and handling. And you can build a fairly complex rules in for how and when that sharing and synchronization is done. We're starting to expand on this concept of there being lots of XNAT systems out there, and that users might want to be able to access data across them using concepts from data federation. So for example, we worked with the Dementia Platform UK, which is building a distributed federated network across all of their imaging centers. So they have, about 6 or 8 different imaging centers spread across the UK that are all imaging dementia cohorts. And they all have XNAT systems set up to manage the data for their particular cohort. And then from a central hub, users can come in and issue queries that are distributed across all the different nodes to find particular data that meet their interests and criteria. They can submit requests to the PIs at each of the different sites, and then get an aggregated data set delivered to them that they can then work with. So this is kind of a bit of a hybrid federation in that the queries out to the data systems are federated, but in the end, an individual data set is produced and pushed to a central hub where the user can work from it. The logic of that is that the central hub can be associated with a large image processing cluster, in which the user can then the data is sitting close to that cluster so that they can do high throughput image processing. We're imagining setting up this kind of federation, not just within a closed network like the DPUK, but globally across all XNAT systems, or at least all XNAT systems that want to participate in a data federation. The idea would be not that data within those systems would just automatically be accessible to all users, but that if an investigator wants to opt in to sharing their data, that summary information about their data set would be published onto the federation. Users would be able to issue queries across the metadata, not the actual data, but metadata. So for example, they could look for across this federation for neuroimaging studies of Parkinson's disease that include PET imaging and MRI, let's say. And then result sets would come back that would allow the user to see data sets of potential interest, to submit requests to the PIs of those data sets. And once given approval, they would then be able to directly access the actual images. The intention of the federation is to enable anonymized views of the data, to make it very straightforward and turnkey to make that data accessible to the community in ways that don't breach the privacy of the research subjects. There's a lot that has to go into making a federation like this work. The technical side, I think, actually is more straightforward than the more research philosophy sorts of issues that come up, and the logistics of how to share potentially sensitive data. But we are proceeding with implementing a lot of the technology, having a lot of conversations with the community about how data sharing in this kind of federation can be done. So one of the things that XNAT supports is analytic infrastructure so that you can run automated processing routines. We use a technology called Docker. So probably most people on the call by now are somewhat familiar with Docker, but the idea is that Docker provides little containers, like little virtual machines, on which you can embed all of the libraries, executables, and other resources that are needed to execute complex computational routines. And those can be all packaged up on one of these containers, and then you can share that container with people. They're very portable. We like the idea of using containers as ways to embed scientific routines that can then be dropped into a platform like XNAT and tied to data and workflows to automate many of the complex computational routines that are used in research. So this is what the XNAT interface looks like for working with containers. There's a user interface that makes it pretty straightforward to configure and identify Docker containers of interest. Under the hood is a JSON specification for exactly what all the fine details of how that container would embed within XNAT. But basically it comes down to, it specifies what's the Docker image of interest. That can be pulled from something like Docker Hub or some other hub that's sharing the Docker containers. And then after that, there's a configuration step to describe what that container is doing, so what kind of operations are embedded on it. And then there's a configuration step, and this is really the kind of the key to getting it to work with XNAT, where you tell it what the images within XNAT or what the files or resources within XNAT that need to feed into the container. And then after the container runs, what did it produce? What kind of files are coming out of it that can be stored back to XNAT? So you might say that this particular container needs the DICOM images for a particular type of scan, and what it's going to produce. In this example, we have it producing NIFTY files. So it's going to run a NIFTY DICOM to NIFTY converter and store those NIFTY files back alongside the DICOM. And then that's really it. After that, XNAT really handles the logistics of executing that container for particular data sets. So from a user interface perspective, for a particular session, a user could come in, and once that container is configured, they would see the command for it sitting right there on the web page. They could click that, and then it would go and run for an individual scan level, as shown here, or here's a blown up view of it in case that was hard to see. Or they could do it at the overall session level, so it would run across all of the scans. This is sort of a manual configuration of this container. It can also be set up to be triggered automatically for different events. So for example, you might want to run the DICOM to NIFTY conversion automatically when the session is archived. And that's just a matter of using XNAT's automation system to configure those kinds of automated executions. So there's a lot of different kinds of things you could build into these kinds of containers. We just mentioned a data format conversion. So for example, going from DICOM to NIFTY. But you can do all kinds of other kinds of computations. You might compute an image histogram or motion histograms. You could transform an image to, for example, register to an atlas, or you could do group level operations for building an atlas. One of the common workflows of a lot of people we're working with is to do deep learning exercises. So you could do take a TensorFlow application and integrate that as a container. So there's really no end to the kinds of analytic routines that can be embedded in these containers and linked into XNAT workflows. Obviously, it's nice because it supports automation and being able to easily share these kinds of analytic routines with your collaborators. One of the things that's of particular interest to me is to really be able to promote reproducible research. And because imaging workflows have tended to be complicated and vary a lot with different versions of underlying software, knowing how to reproduce someone's image processing results can be difficult. And I think being able to share containers will really make that a lot easier, because the complexities of the exact libraries and versions of software tend to go away. So that's kind of the overall feature set of the XNAT system. One of the main use cases that we've been using it for is for data sharing. And this is kind of how we often think of the data sharing workflow, that initially images are managed within an XNAT system that's private. So data coming from the scanner or PACS goes into an XNAT system where your researchers have access to it. And at some point, once they are ready to share the data, they actually move it to a secondary system where they can be more open with sharing the data. There's often a selection process where a subset of the data is made available, and potentially various kinds of anonymization or recoding of the images and data are done before moving it to the more public system. At the public side, there's often still user authentication that's required. Users still have to sign data use terms. And it's not just because it's been moved to this public system, the data is not necessarily just automatically available to everyone. There is still an approval process. So we've implemented this kind of data sharing for the Human Connectome Project, and have shared data for quite a while now, starting as early as 2012. We've been sharing data sets out of the Human Connectome Project, including very large amounts of high resolution functional and structural imaging. We've now finished collection of data within the HCP and have made it all available, using both internally what we call the intra-DB system for managing the data as it's being acquired and as we're doing internal workflows, and then through Connectome DB, which is the public-facing XNAT instance through which we make the data available. Here's kind of a detailed view of what that overall workflow looks like. And at this point, we've shared it with lots and lots of users. So over 15,000 people have created accounts on the Connectome DB system. That includes many, many users, not just in the US, but around the world. To date, the US has downloaded and used more data than any other country, but China has been rapidly catching up. And we've been sharing data for quite a while, and the data utilization continues to be very high. We have spikes around data release times, but even outside of that, the data access has been fairly sustained. This view is just of users downloading the data through our own web portal. They've downloaded over 8 petabytes of data over the last 5 years. We're starting to see a little bit of a drop-off in people accessing data through this resource. And that's partly, well, I'll tell you why that is in just a second. We also have been shipping these data in what we call Connectome in a box. So if you're at a site that it's really hard to download really large data sets, it's not necessarily practical to try to do it over the web. So we have been shipping hard drives. This is a terrible, terrible thing to do. It's been super painful for us. But for many of our users, it's really the only practical way to get the data. We encourage them, once they have the hard drives, that they make them accessible to others at their institution so that they aren't all separately ordering their own copies. But we've sent out several hundred of these by now, both domestically and internationally. But the main reason I think that the downloads are starting to drop off is that we do have the data accessible on the Amazon cloud through the Amazon's public datasets program. And so users can just come to our website, sign our data use terms, and then get submit their AWS credentials to get a key that allows them to access the data on the Amazon cloud. We've had over 1,000 users who've gotten their AWS credentials to access the data this way. And I think it's turning out to be a very popular way to get to the data. And it's supported in part by the fact that all of the Connectome pipelines have been made available through the NITRIC EC2 instance that can easily be launched on AWS, and then users have easy access to all of those tools. As I mentioned, this is done through the AWS Public Datasets program, which is something they're offering now to share at no cost to the data providers storage of that data. Obviously, to do computation requires downstream users to pay for the computational resources on the cloud. So HCP obviously is one of the high value data sets that are out there, but there are many others. Here's just a few. I won't go into any detail on these because we're starting to run a little short on time. In addition to the data sets, there's a lot of data repositories that are out there that have long lists of data available across a number of domains, from cancer imaging, neuroimaging, and others. So I'm going to very briefly, just for a minute or two, talk about some of our translational systems that we've set up. And there's really kind of two workflows. One is if for a particular individual patient, you want to run some analytic routine that was developed in the lab and explore how useful that is on the clinical side. The other is, and this is starting to get to be very popular, extracting very, very large data sets from the clinical packs, largely to feed into deep learning systems that require very large data sets to train their algorithms. So we've started to set up infrastructure to do these kinds of workflows. So this is a system we call the TIP that allows users to query out to the PACS, find a case of interest, run one of the Docker containers that includes an analytic routine, generate some sort of DICOM files from that container, allow a user to do quality control before pushing back to PACS. If the processing passes quality control, then it could go back into the PACS. Obviously, this kind of communication requires work very closely with the clinic and with the IT environment in the clinic to approve this kind of workflow and the kinds of images that are being pushed back to PACS. But it can be a very nice way to quickly translate research capabilities into the clinic in a controlled way. Really, the key is this step 3, where the quality control is done, because that's what gives the assurance to the clinicians that what's being produced and pushed back to PACS is suitable for clinical review. We've done this with a brain tumor protocol that's using resting state fMRI to identify the functional networks around brain tumors and to help guide a neurosurgeon to remove the tumor without interfering with key functional areas such as language and motor areas. When eventually, as the cases gets processed through our system, it gets pushed, the maps we generate of those functional areas are sent to the surgical navigation system, which is shown here, and then that's registered to the images and co-registered stereotactically with the patient during surgery, so the patient can see precisely in registration with where they're actually resecting tumor exactly where the functional areas are. Very quickly, these large patient cohorts, we've started to get into pulling these kinds of data sets that can exceed, hundreds of thousands of exams. So for example, we have a study going here of looking at CT and stroke that has hundreds of thousands of CT exams. And we're finding, some various logistical challenges with doing that. A primary one is actually interfering with the PACS. The IT services are concerned that if we try to pull hundreds of thousands of cases as quickly as we can, that would interfere with clinical use of the system. And then once the images are moved into our system, there's a lot of challenges with I/O throughput. The deep learning tools that are in place now depend on GPUs and many, many thousands of cores, and those cores are very, very hungry for data and will very quickly throttle your file systems. So that's something that we're working on now and finding to be one of the kind of biggest challenges in dealing with the kinds of data sets that are being used in deep learning tasks. Maybe I'll cut it here because I think we only have a few more minutes, and I know that Jack wanted to have a few minutes for some questions, so maybe I can just cut it off here, and I'll have all these slides available so that the ones I didn't get to, people can just kind of look at on their own time if they're interested. So Jack, I'll hand it back to you.
Thank you so much, Dan. That was a really great overview of what is a very impressive system, and it looks like it's being applied in many clever ways that will be of interest to many of our attendees today. One question I had for you, which looks like somebody was asking about, is where can they find these great tools? Can they find these on your website, or how should they look for them?
You can. The XNAT system is available at xnat.org, so that's really easy. Some of the other tools that I mentioned, you can usually just search on those in Google and they'll pretty easily come up, but the XNAT stuff is all at xnat.org. Fantastic.
And one other thing that occurred to me is, while you've really talked about the imaging informatics, increasingly, in addition to the imaging, are all kinds of associated data, other things like genetics or full genome sequencing, maybe proteomics or microbiome related data, for example, associated with each subject who's also undergone human neuroimaging or medical imaging. Is XNAT, does it have the facility to incorporate that information, or is it linked in some way? How does it handle that?
It can kind of be one or the other. For kind of simple metrics that might be extracted from those kinds of complex data sets. Those are easily stored in XNAT and can make querying for data sets easier. But for the actual full data sets, it's often the case that a system that's really tailored to that kind of data, so genomic database, is more appropriate for handling and storing and managing workflows around that particular data. And then you can do your aggregation in a number of ways. The primary way we do it is using the API and various Python libraries. So we have a library called PyXNAT that allows for easy querying and searching and pulling data from XNAT in Python routines. And there's for many of the other kinds of databases, so maybe like a REDCap, for example, there are also Python libraries for that. So you can use Python as your environment for querying across systems and pulling together and linking data sets. Usually you're using coded identifiers for your research subjects and their visits, so that those become the key to linking between data sets.
Okay, fantastic. Well, once again, thank you so much, Dan Marcus from Washington University, for taking part in our BD2K Guide to the Fundamentals of Data Science webinar series. I want to thank everybody who joined us today. Please do use your favorite search engine and look up for more information on XNAT and its associated tools. Thank you, everyone. Have a great weekend, and we'll see you next time. Thank you so much, Dan.
Bye. Thank you, Dak, and everybody.